record button because I have been known to forget that, and that's never a good thing. Okay, we have straight up 12 noon, and I'd like to welcome everybody to the What is the Impact of Inbreeding in Holstein Dairy Cow webinar through the Iowa State University Extension Dairy Team. Today we have with us Dr. John Cole. He serves the Council on Dairy Cattle Breeding as a Chief Research and Development Officer. Before coming to that organization, he spent 3 years as a Senior Vice President of Research and Development for Peak Genetics. Dr. Cole has estimated annual losses of at least $10.7 million due to known recessives in the dairy industry. Recently, when they were reviewing the calculations for the April 25th or April 2025 run, their organization, the NAAB Sire Evaluation Committee, noted what seemed to be smaller than expected base adjustments. The investigation by those two groups of researchers found that these differences were due to larger adjustment for inbreeding because of the rapid increase in the relationships among young animals over the past 5 years. Dr. Cole will share his insights on this and make recommendations. Dr. Cole, as I always say, the podium is yours.
Well, so thank you. I appreciate the kind introduction. And maybe I'll add, before I was working at CDCB, which is the National Genetic Evaluation Center for U.S. dairy cattle, and then working at Peak, I was also with the USDA Agricultural Research Service for 17 years. So many of the things I'm going to talk about today reflect the contributions of my colleagues and collaborators. And I don't want folks to think that every single thing I talk about here is something I figured out, because I try to always work with folks who know more than I do. So I'm going to talk about Holstein cattle today, but I want to make the point that a lot of the general things I say about inbreeding are going to be similar both in other breeds of dairy cattle, and then say if we were to look at beef cattle, or we were to look at Indocene instead of taurine cattle. So I will show some specific figures from the Holstein breed, but this generalizes beyond that. So let's see, let me get in the right window. So I'm going to talk about sort of grouped my comments into a few different topics today. The first thing I'm going to do is sort of talk about what inbreeding is. And sometimes if you ask one person, they'll tell you something different than another person. So I'm going to try to give us a common vocabulary for talking about it. I'll talk some about why new inbreeding or recent inbreeding matters more than old inbreeding. I'll talk some about how inbreeding affects different traits. I'll share some ideas that we've talked about in the industry for how we might address the problem, at least in the dairy sector. I'll have a little section towards the end on genetic defects, and then a couple of concluding remarks. So, the first thing really is what is inbreeding, right? Inbreeding is a natural consequence of mating related animals in a population of a fixed size, right? There are only so many, 9.3 million dairy cows in the U.S. So everybody's related in some way or another. And sort of the historical definition that we use is, when you talk about inbreeding as like a coefficient of inbreeding, it's the proportion of an animal's genome where haplotypes are identical because they came from the same ancestor. And as I said, it comes from the mating of related animals. Now, the reality is that inbreeding is inevitable in a population of a fixed size. So the accumulation of inbreeding, it does not necessarily represent mistakes made on the part of breeders. It's simply a property of genetics in a population. So we can manage inbreeding, but we cannot completely avoid it. And I like to share with you here, this is on the right is a pedigree from a Holstein cow in the Beltsville Agricultural Research Center herd back in the 1940s, where they did a series of inbreeding experiments with Holsteins and Guernseys. And this is about the most inbred Holstein cow that I know of. She was about 65% inbred. Not quite as inbred as the Hereford cow used for the reference genome, right, as Dominette was, but pretty impressive nonetheless. And this was the result of several generations of intensive mating, including daughter offspring mating. And if we were to rewrite this pedigree, there aren't a lot of pedigree doesn't fork a lot. Now, today, like I say, not today, classically, we talk about inbreeding calculated from pedigrees, right? That is, we know the parents of an animal, we know the parents of those individuals. In the US genetic evaluation system, we trace pedigrees back to 1960. We do have some pedigree data that go back further, but 1960s are base. Now, an important thing about this is that this assumes when we calculate inbreeding from a pedigree, it assumes an animal receives exactly half of their of their genes from their sire and dam. Well, that's pretty much entirely correct. But if you go back a generation, you don't get exactly 1/4 of your genome necessarily from each grandparent. If you step back to great grandparents, you don't necessarily get, exactly an eighth, etc. But as I said, this is the proportion of alleles identical by descent. And I showed this to a graduate seminar at Davis earlier this year. And this is from my undergraduate genetics book. I had to draw numbers and arrows and try to make sense of it. But sometimes it's nice to see where things came from. But in about 2008, we ended up with a new way to trace relationships and inbreeding in the population, which is by using genomic information from high density DNA marker panels. So instead of assuming that an animal got, say, 1/4 of their genes from each grandparent, an eighth from each great grandparent, etc., we can actually look at their genome and see precisely for each chromosome exactly what proportion of relationship they have with each animal back in their pedigree. And this is an example here. It's chromosome 24 from a Holstein bull named Megastar. And, it's showing you, if we go back to the sire is on the top, the dam's on the bottom, and you can see, for example, that he has the bull Aerostar on both sides of his pedigree. So you can see this particular chromosome, he inherited almost all of his chromosome 24 alleles from Aerostar and virtually none from the other animals in the pedigree. Now, you might be saying that's probably not typical, and that's true. We sort of went and cherry picked this example. But the point that you'll see in the parental generation and the grandparents is that we can track the haplotypes and we can track recombinations. So we can very precisely calculate relationships and inbreeding. One thing I will caution folks is if you go, for example, to the Council on Dairy Cattle Breeding's website and you look up the information about a bull, you're going to see a pedigree inbreeding measure and you're going to see a genomic inbreeding measure. The genomic number is pretty much always going to be bigger because it's actually, it considers genes that are, I'm sorry, alleles that are identical because they came from the same ancestor, as well as alleles that are identical or that are homozygous just by chance. So you're actually measuring slightly different things with the two measures. Some folks, if you listen to us scientists talk, you will also sometimes hear about something called runs of homozygosity. And that's basically you can look at the genome using DNA marker information And you can look to see, okay, how much of the genome is in long, unbroken stretches of DNA? How much of the genome is from smaller pieces of DNA? And you can use that to sort of determine how old the inbreeding in an individual is, right? And regions that where inbreeding occurred a long time ago, say maybe in the 1970s, there will the homozygous regions will be very short because there have been many generations in between. There's been lots of recombination. Recent inbreeding will produce long stretches or runs of homozygosity. The thing that runs of homozygosity tell us is they can help us look at specific places in the genome if we're trying to really dig down into the biology of, say, where inbreeding depression or conversely where hybrid vigor might come from. And we're also interested in old inbreeding because those regions have typically been purged of harmful alleles. That is, the harmful alleles have been eliminated from the population and the desirable alleles have been maintained. I'm going to come back to this in a little bit. And ultimately, we talk about inbreeding and we're concerned about inbreeding because it can have undesirable effects in the population, right? So if you think back to your undergrad genetics and they say, hey, you don't want to do a lot of inbreeding, um, inbreeding does not produce genetic defects, right? It is not causal. But if you do lots of inbreeding, there's the possibility that you'll uncover existing genetic defects. So if you think about Holsteins, for example, maybe you think about something like Holstein haplotype 1, which is a deletion in a gene called APAF1. There was a bull that was heavily used where that mutation arose. And as you do more inbreeding to that bull, you increase the likelihood that you'll have two copies of the undesirable allele in the population. HH1 affects fertility, causes early embryonic loss, so you see a decrease in fertility. And we think that these fairly simple Mendelian effects account for most of the effects of inbreeding depression. And if you inbreed slowly, you can sometimes kind of do artificial purging, but you have to know about a defect to manage it that way. And most of the time, I told you that inbreeding is not causal. Inbreeding doesn't make mutations happen. That's true. What inbreeding does tend to do is if you have an undesirable allele located physically near a desirable allele, as you do selection in the population, the undesirable allele gets dragged along with the one you're selecting for, and we call that hitchhiking. So, why does selection favor inbreeding? This is probably the most geneticsy, right, that I'm going to get here in terms of talking about the statistics of it all, because our dairy cattle, the intensive selection we do in dairy cattle actually has some characteristics that favor the buildup of inbreeding. The first is that we do selection over multiple generations, and we do intense directional selection. That is, we're trying to move the population average in a certain direction. So most folks think about milk production in dairy cattle, but these days, in most countries, there's a complex selection objective that involves a number of traits, not just milk production. So fertility, longevity, health, etc. There's also a lot of variation between individuals in their reproductive success. And what that's really saying, right, is that very successful dairy bulls can produce lots and lots of offspring, thousands of offspring, tens or hundreds of thousands of granddaughters potentially in a few cases. And so that high variation in reproductive rate drives us towards family-based selection. We use a statistical methodology called best linear unbiased prediction to calculate genetic evaluations. And BLUP really pushes you towards within family selection. So as you have a family that's successful, because you have a lot of information about that family, you tend to keep selecting within that family. And that means at the end of the day that we tend to use lots of related animals as the parents of the next generation. And if you go look at the genetic evaluations every 4 months, you'll look and see at the top of the list, you'll see a lot of animals that share the same prefix, that have the same grandsire or sire. And so that really pushes us this way. Sometimes inbreeding is deliberate, right? So I'm from the dairy cattle perspective, mostly we don't really like inbreeding because it has negative consequences. But you can think about a lot of terminal crossing systems, say swine or beef cattle. They will do some inbreeding, poultry breeding. The crop folks do a lot of crossing of inbred lines. And sometimes you're trying to fix phenotypic characteristics more than performance characteristics. I also mentioned horses down at the bottom because I think everybody talks about it, right, when it's Kentucky Derby time, about how inbred all the horses in the race. I think they probably all have very similar pedigrees if you go back and look. So it's not something that only happens in dairy cattle, and it's not something that strictly happens by accident because we don't know better. So that's just trying to give us a little background on what inbreeding is. And so now we're going to get into, say, some characteristics of the US dairy cattle population. So here we're looking at proven genotyped bulls. So these are genotyped Holstein bulls that have daughters who have generated their own performance information. And I went back to 1980. I didn't go all the way back to 1960. Things are pretty flat before 1980. And you can see here that the solid line is pedigree inbreeding. The genomic inbreeding is sort of the long dashed line, and the short dashes are the genomic future inbreeding. And you'll see here, right, that they're all increasing. And the genomic future inbreeding has really been increasing a lot since the start of genomic selection. And again, this is often tied to the way we do breeding in the population. You'll see that if you go look at it by stud, for example, you'll see that the AI companies often produce a lot of sons of a successful bull, trying to find the next outstanding bull. You'll see that, say, there's lots of sons of Captain and Charles out there. That's an ST Genetics bull. If you look on, say, at ABS, ABS had a bull named Mendel P, who was a homozygous polled bull, which is not that common in Holsteins. Lots of Mendel P sons in the population. And so we're really pushing these inbreeding rates up pretty fast. And this is just an example bull. Here, I'm not trying to pick on anybody. This is just, I talked at Holstein Association a few years back, and I use this bull as an example because you'll see the same animals popping up on both sides of his pedigree, especially this inbreeding to Robust and Shauna as well. And you're sort of double inbred. You're inbreeding to two different lines through both sides of the pedigree. At the time, when I put this slide together originally, this was the most inbred bull in the Holstein breed. And you'll see here his pedigree inbreeding is almost 13%. His genomic inbreeding is about 15%. His net merit $776. That's a little unfair to him because we just had a base adjustment. So his inbreeding, I'm sorry, his net merit, which is our economic index, was higher before the base adjustment. And his TPI is still over 3,000. This is a good bull. He's made lots of sons. But this is something that I would, as a geneticist, I would feel a little hesitation about. I wouldn't want to see this in every pedigree. And as I mentioned, a few slides ago, this is not a problem only in dairy cattle. So this is some work we did. I did with some colleagues at North Carolina State looking at Angus cattle in the US. So you can see whether you're looking at pedigree inbreeding on the top, genomic inbreeding in the middle, or runs of homozygosity on the bottom, you see similar trends over time, much more intensive inbreeding in that population. Won't spend a lot of time on this. It's not just a cattle thing. This is some data from commercial rainbow trout lines, right? So if you're looking at the aquaculture side, you see it. my PhD work was with working dogs. So this is showing something similar in dog guides, right? So both in German Shepherd and Labrador Retriever dog guides at the CNI. So both the relationships and the inbreeding are higher in the German Shepherds than the Labs, but not really attractive rates of inbreeding there. And so why do we really care so much? Well, inbreeding is tricky, and I'm reminded of an interview that was once done with Ernest Hemingway, and he was asked how he went bankrupt, and he said gradually and then suddenly. So you're wondering, well, what does that have to do with this? The challenge with inbreeding is we really don't know how much is too much. What we know from studies in model organisms, so by that I mean, say, species like flies or mice, where you can do really, really fast turnover of generations. So you can see, well, what if we make, a 95% inbred fly? What happens, right? And I know you're thinking flies aren't cows. The problem is everything looks great until it doesn't, and the population can sort of cross a threshold and then crash very quickly. So we worry in part because we don't know when we're going to go from having a manageable situation to potentially having an unmanageable situation. Now, on the other hand, so I don't alarm everyone who's listening, we also don't do single trait selection anymore, right? When I was an undergraduate, what were the traits available for dairy cattle? You had linear type traits. You had milk, fat, protein, somatic cell. Well, somatic cell score came along when I was doing my master's, so we didn't even have that when I was an undergraduate. So you basically had production traits, and that was it. So all you have is a hammer. Everything looks like a nail, and single trait selection can get you into trouble very quickly. That is not how we've been selecting cattle for since 1994 in the U.S., right? That's when the first version of lifetime net merit came out, and you see similar use of total merit indices in other countries. So what we don't know is if we're going to see just a slow, steady erosion of performance over time as inbreeding depression builds up, or if we're going to cross a threshold and see a sudden, rapid, and dramatic decrease in performance. And what if you're wondering what's in this graph here? Along the x-axis is the bull's breeding value or his predicted transmitting ability for daughter pregnancy rate, which is a measure of fertility. And then on the y-axis is the inbreeding of the individual bull. And you can see it's not the correlation is not one, but it's definitely negative and moving in an unfavorable direction. And how much of that can we sustain? And so here I'm trying to show some examples of how inbreeding affects different traits. This is from a paper that one of our colleagues in Europe did, Leroy, in 2014. Now, the thing here is that he standardized all of the traits. Basically, you basically standardize it to a mean of 0, standard deviation of 1, and then you can do a regression on inbreeding. The point is that means you can make comparisons of one trait against another. I'll show you some numbers from the US population on the next slide, and I'll explain why you can't always compare those directly. But here I looked at several traits. I just pulled some things out of his paper because he looked at a lot more traits than this, but you can't read the table when I put it on a slide. So yeah, here I'll do fertility, survival, body weight, right? Some yield traits, somatic cell count, and locomotion. And what you see here is that in every case, the effect of inbreeding is harmful on performance. So as inbreeding increases, performance for that trait decreases. It's it does vary in intensity. It's not so big on fertility. But it is more substantial for, say, survival or some production traits in the data he looked at. Okay, so we don't like this. This is a problem. What are we going to do? Well, so this is the general picture. Now, I'm asked sometimes about should dairy producers in the U.S. worry about inbreeding? And so here's how I try to answer that question. There are two tables on this slide. The top table is from Council on Dairy Cattle Breeding, and we publish these numbers every 4 months when the new genetic evaluations come out. And this is the change in phenotypic performance from a 1% increase in inbreeding. So I'll give you an example. If you look at the top row, if inbreeding goes up 1% in the population, on average, you lose about 74 pounds of milk. You know, you lose almost 3 pounds of fat, a couple of pounds of protein. And this information is available for more traits than I'm showing you here. But if I put too much on the slide, again, it's hard to read. And these are, I believe I updated these numbers in April after the most recent evaluation. The second row in the top table that has the blue rectangle around it is the change in phenotypic performance when inbreeding goes up by 1/4%. And you're like, well, why 1/4%? Because that's roughly the rate of increase of inbreeding in the Holstein population each year right now. And I did that so we can then compare it. So then you'll see instead of being 73.6 pounds of milk, it's 18.4 pounds of milk. I did that so we can compare it to the bottom table, which is from our colleagues at the USDA Animal Genomics and Improvement Laboratory. And this is the change, the correlated response to selection from using the lifetime net merit index. Okay, so here you see in the bottom table, each year we expect to see inbreeding, we expect to see a gain of 127 pounds of milk simply because of the improvement in genetic merit. We're going to lose about 18 pounds from an increase in inbreeding depression in the population. So it's a net gain of about 106 pounds. And so I looked at this across some of the traits we're interested in. Green shows you everything is good. The genetic trend is higher than the inbreeding depression until you get to daughter pregnancy rate, which, as a reminder, is a measure of fertility. Well, our inbreeding depression is is decreasing, right? So we're losing ground faster than we're gaining ground from genetics. Now, I would point out this is not a uniformly bleak situation, because if you look here, we also have heifer conception rate, which is HCR, and cow conception rate, which is CCR. And we're actually making better genetic gain for those traits than for DPR. I don't have time to get into it today. DPR is probably not the best thing to look at when you want to talk about fertility anyway, because some changes in management practices in the dairy industry have sort of gotten ahead of how DPR works. So to answer the question, I would say folks don't need to worry about it, but that's not the same as saying that people should ignore it. I mentioned when I defined runs of homozygosity as well, that we can use, and this is some work that my colleague Christian Malteca did at NC State, and you can actually look for places in the genome where inbreeding depression harms performance, but it doesn't result in, say, in the death of an embryo or the death of a fetus. So, and these are actually sort of tricky. And we're hoping, the point is we're hoping to use this information that we get from runs of homozygosity to better understand the biology of inbreeding depression in the dairy cow. And shown here, I know this is, I'm not going to really spend a lot of time on how to interpret this chart. I think the point is, if you're wondering what are the researchers doing about this, we're doing a lot of work to try to actually go from a statistical understanding of inbreeding to a biological understanding of inbreeding. So what do we do about inbreeding then? You know, we see that it's happening. We see that it has undesirable effects. So what's the solution? Well, there's a lot of different things we could think about doing. And in fact, this is something that we currently do in the US today. When we calculate the US genomic evaluations, we actually adjust the PTA of every bull and cow to account for inbreeding they're expected to produce in the future. And in fact, we adjust the PTAs already. Here's what I will tell you. The PTA adjustment for most animals um, doesn't move them very far. So if a bull is really, really good, even after you adjust his PTA for inbreeding, he's still going to be a really good bull. And people are still going to want to use that bull heavily. So it's a strategy that we use. I don't know if it's really helped very much. We also use genomic inbreeding instead of pedigree inbreeding in that. And there are other things you can imagine doing. For example, limit the number of active bulls that are permitted from a sire family. And you could also think about, hey, could we potentially limit the number of embryos produced by elite heifers? Because bulls will always make lots and lots more offspring than individual cows, but using some of the newer reproductive management tools like oocyte pickup, there are some cow families that are now producing a relatively large number of offspring compared to the past. I already showed you guys this pedigree. Well, you know what? I told you that in the US, we trace our pedigrees back to 1960. So that's where we start calculating the buildup of inbreeding. Well, if we were to change that from 1960 to 1970, made 1970 our 0 point, well, a bunch of inbreeding would go away. And if we moved it to 1980, even more inbreeding would go away. Now, why would you do this, right? I mean, someone's thinking, well, that's a trick and that's not really useful. Well, what that does do when you shift your reference point is it focuses more on recent inbreeding than on historical inbreeding. Because if you leave that point, say, fixed at 1960, every year you move into the future, inbreeding is going to the average inbreeding in the population is going to increase. Some countries do already adjust their pedigrees for this. Some examples are Italy, Slovak Republic, the UK. So some countries trim the pedigrees. So for example, they only go back 4 generations or 3 generations. Other countries change their base year. Pros and cons there. It is difficult to explain to people. I proposed at a Discover conference a couple of years ago that maybe we just shouldn't publish the PTAs. If we don't publish the breeding values, then folks can't go all try to use the same bull at the same time and continue to heavily go into these elite families. So how would this work? Well, I don't know exactly how it would work. Maybe you would just mate your cows at random to the bulls active in the population. Maybe you could have some sort of system where you say, green is a good bull in a particular trait, yellow is average, and red is maybe less desirable. So you would sort of band the animals instead of showing you the exact PTAs. Or maybe the software knows what the PTAs are, but you don't. So when you're making some selection decisions, you're not always steered to the same elite bulls. I bet for the folks, if some folks have joined us from various farms around the country, they're probably not super excited about this idea. You could think about, as we look at the dairy population and we see much higher rates of embryo transfer than we've seen in the past, you could say, well, what if we just use terminal dairy embryos and then we don't necessarily care about the inbreeding of the cows in the herd because they're not the animals producing the next generation, and then you could manage your inbreeding in the nucleus herd where the embryos are made, right? So you wouldn't really have any inbreeding the way we're used to thinking about it today, but someone still has to maintain purebred lines, and that's something I don't think I have a slide in here on crossbreeding, and that also comes up with crossbreeding, right? Folks sometimes say, well, can't we just use crossbreeding? Then we don't have to worry about inbreeding because they're unrelated. Someone still has to maintain the purebred lines. And I would also say that I don't think that today it's very reasonable to say that we're going to breed every dairy cow in the US by embryo transfer, or at least that we're going to breed every cow that we want a replacement from by embryo transfer, because doing embryo transfer is harder. It takes more skill than doing AI, and you're going to have to build that cost into the embryos, and all of a sudden the price may not be that appealing compared to just breeding by AI. As a geneticist, maybe someone says, well, hey, can't you guys do some math and change the selection index so that it thinks about inbreeding in some way? And this is certainly possible. You can add measures of genetic diversity into your selection index. Um, when you do that, what you're really doing is you're doing something called optimal contribution selection, which everyone in the US has very carefully ignored since it was first proposed. It's got to be maybe the late 90s when our colleagues at Edinburgh first proposed it. The reason folks often don't like optimal contribution selection is you give up short-term genetic progress to maintain diversity in the population. Over long periods of time, you make about the same net selection progress, but it's simply something that doesn't seem to have resonated well with a lot of people. They just don't like the idea of giving up those selection gains. Now, today, we have new high tech solutions, and some folks will say, well, why don't we use some of this gene editing stuff, right? Can't we use that instead? Then we don't have to worry about inbreeding. You can just go in there and make some tweaks to the DNA, and then there's no bad inbreeding. And that was proposed. Some of our colleagues at University of Edinburgh did some modeling on that. Um, if you can make something like surrogate sires work, then you don't even have to lose a lot of time, so you don't fall too far behind on the genetic trend. And then you can have lots of genetic gain and no bad consequences. The single biggest problem with this is that we don't know where it's okay to have a lot of inbreeding in the genome and where it's really important to maintain genetic diversity. I know we have some veterinary students on, and they might be thinking, oh, but that's not true. You want to have lots of diversity, right, in the immune gene complexes. And that is true. I agree with that 100%. Our reference assembly is fairly poor in those regions in the cattle genome, and we don't have a lot of good markers there to do selection. So this sounds great, but it's actually pretty challenging. The other thing I haven't really mentioned is the animal with the most edits that I know of. I believe there's a calf on the ground that has 5 multiplexed edits. Now there could be someone who's done more, because I don't know what every company is doing. The point is, to make this work, you're really talking about doing genome editing at many locations in the genome at a time. We don't really are not able to do that yet. As we continue, as each our AI company continues to develop their own families that they're doing selection in. And I'd like to point out that we used AI first. So when I say AI here, I mean artificial insemination, not the other AI. And so some folks have said, well, hey, I'm going to use ABS bulls for a while, then I'm going to use select bulls for a while, and I'm going to sort of cross between these populations, almost like you're doing rotational crossbreeding, but you're crossing purebred lines from different studs. And I think there is some data, some work that Tom Lawler from Holstein Association and the folks in Ignacy Mistel's group did. Yvette Stein did some work at University of Georgia on this. There is some evidence that says that the different companies are selecting for different things. I don't think that's a surprise. I don't know how much variation we could expect to recover from this, but it is something that folks have thought a little bit about. And I suppose one of the other things here is there's this technique or this concept called in vitro breeding, where you would basically take stem cells, you would culture those stem cells, and you would force them to differentiate into oocytes or into spermatozoa. And you can do several generations a year of selection on the bench before you ever have to make a live animal. And the idea here is that you could do very intensive selection at the bench. You could then sequence and screen those lines and determine which ones are free of undesirable genetic effects, and use those to produce live animals for the next generation. It's very, very elegant. It doesn't work yet. I'll be honest, I'm kind of skeptical that it's ever going to work, but maybe I'll be proven wrong. Um, but there are a lot of ideas like this that our some of our colleagues have dreamed up, and we'll see what happens there. And finally, this is not always the most popular thing, but I think it's important for us to face this and think about it. Okay, we are given a license to operate by consumers, right? That is the folks who go to the store and spend their money to purchase the food that farmers make. And if we don't do something to get a handle on inbreeding and relationships in the population, if consumers come to perceive that that this is essentially a welfare problem, they may choose to not continue to purchase dairy, for example. And what we have to remember is that it's true that most folks don't understand what happens on farms or why farmers do what they do. And maybe that's not fair that folks cast judgment, but this is a reality, right? And I can tell you, not in the US, but in other parts of the world, some of these questions are already being asked, especially when you want to go to your banker and you want to get a loan, you want to expand, you want to build new facilities. You're getting questions like this. When I was working for Peak, this Wall Street Journal article appeared, and I had to answer a lot of questions from the owners of the company about what the heck is going on and why is this in the front page of the newspaper. So we need to consider this when we say, well, I'm not going to do something about inbreeding. I'm going to let somebody else do something about it. Now, I know a few of the things I've suggested as possibilities do seem a little far-fetched, and so some of you might be saying, is this guy serious? Well, first, I'm going to remind folks that genetic evaluation, right? That's what I do. That's what Council on Dairy Cattle Breeding does. Genetic evaluation is a tool to rank animals for selection. And we then do selection to change characteristics of the population. Genetic evaluation is not actually a tool for managing genetic diversity. So we already do some adjustments that I talked about to penalize animals that produce a lot of inbreeding. What I probably didn't state clearly is that animals that are less inbred than average actually get a little bonus added to their evaluation. Now, these are ad hoc. There's no theoretical justification for doing them, really. And the truth is, I'm not exactly sure that they're really going to achieve what we want to achieve with them. So this might make us feel like we're doing something, but I don't know if we're really actually doing something. And that goes back to my previous comments about the New York Times article. At some point, we're going to have to actually do something, not just say we're doing something or look like we're doing something. And look, there are a lot of solutions, to managing inbreeding. In fact, maybe there aren't there are, I won't say dozens plural, but there are a lot of ideas published in the scientific literature. Many of them have a very elegant theoretical foundation, and nobody uses them. The geneticists don't breed cows. I know some of my my colleagues in the industry argued with me about this, but I pointed out that at the end of the day, dairy producers tell their genetics consultant what sort of bulls they want to use in their population. But they're the ones ultimately driving the decision to select and use certain bulls. Now, many cows are now mated at random. I'm sorry. Yes, cows are now often mated at random to groups of bulls as herd sizes increase and as labor becomes more expensive. Um, Cows don't get individualized mating attention like they might have done in the past in all herds. Obviously, you will find herds where that still happens. A particular bull is matched up with a particular cow. You go to some larger dairies, that's not what's happening, right? There's 1,000 cows to breed today, and no one's going to stand there and go down a sheet and pull 50 different straws of semen from 50 different bulls to get in the right cow. And everyone is absolutely convinced that this is a problem their neighbor should solve. And I told that to the folks at Holstein Association, and I saw a lot more people nodding yes in agreement than shaking their head no in disagreement. So we have optimal contribution theory. You know, we can minimize progeny inbreeding. We can use linear programming to do mate allocation. We can do look ahead mate selection. All these different ideas for how to manage inbreeding. But if nobody uses them, it doesn't matter. And now I see I need to make sure I stay on time here. So I'm going to wrap up with some thoughts here on genetic defects. And this is just a reminder for everyone, right? I'm going to be talking about recessive defects and following the very classic model of Mendel, right? So we have a single single locus, we have two alleles. I won't get sidetracked. There's some cool stuff just done about Mendel's experiments recently. But the point is, we're looking at a very simple case here, simple genetic defects. I'm going to skip over this, but some folks just figured out the last couple of Mendel's P traits biologically. So if you look at US Holsteins, for example, you're going to see we have what sure looks like a lot of known genetic defects, right? And folks are maybe concerned about this. And the good news is we now know the mutation in DNA responsible for each of these, and there are genetic tests now available for each of them, direct genetic tests, instead of having to rely only on a haplotype call. And I recently was curious about, we did this in a paper several years ago, so I updated the numbers. How many genetic defects do individuals carry, right? Are we stacking these up so that we have individuals that have 3 or 4 or 5 genetic defects for which they're a carrier? Well, in AirSures, you don't see that. Part of that's a function of the population size. The more animals, the more likely you're going to get 1 or 2. You see in brown Swiss, there are a couple, both a few bulls and a few cows that carry 3 known defects. Holstein, I thought this was actually pretty good. There are 5 cows at the time I did this, which was in April, that were carriers of 5 known genetic defects. There were 2 bulls and 164 cows that carried 4, and the numbers go down from there. Jersey, again, the population is not big enough. You only see 2. So I thought this was pretty good. Why? Because we're doing really intensive within family selection, but we're not seeing a lot of animals who are carriers of lots and lots of defects when you consider the population size. I don't get to take credit for this. A lot of this is driven by the genetics companies who are like, our customers don't want this. We're not gonna market semen from bulls that carry lots of defects. But just wanted to know where we stand. How do we validate these variants, right? Because sometimes we know that we've got a problem, but it's hard to prove if a mutation actually causes something. This is expensive and challenging to do in large animals like cattle, but if a genetic defect acts early in development and causes early embryonic loss, we can actually do gene knockouts in embryos, and then we can screen those to confirm the causal variant. So this is looking at HH2 in Holstein haplotype 2. Our candidate is in a gene called IFT80, And that's involved in Wnt and hedgehog, which are two important signaling pathways in growth and development. When you disrupt those pathways, you see all sorts of problems. And so maybe folks will take my word for it. I'm not an embryologist. I worked with some embryologists who did the knockouts for us. So in the top left photo, you're going to see control embryos. In the bottom right, you're going to see the knockout embryos. So this photo in the bottom right, I'm told that these are degenerate, dying embryos, unhappy. So when you break the gene on purpose, the embryos all die. When you have wild type embryos that are not homozygous, you see normal growth and development. So we developed the gene test, and you can use a SNP to track the mutation now. When we identify these genetic defects, we're able to manage them effectively in the population. So you'll see here, this is a number of different defects. It's mostly Holsteins, but you'll see Jersey haplotype 1 crept in on the bottom. The red line in each plot is showing you when we discovered the haplotype. So you can see that once we had a good way to track these defects to let cow owners and bull owners know when their animals were carriers, these frequencies decreased pretty rapidly because folks were able to manage these defects because they had information about it. So inbreeding is inevitable. And the truth is, you're always going to have some genetic defects in a population, even if you can't see them, even if you don't know they're there. When you can detect them, you can manage them easily. And it was mentioned at the beginning, we did some work in 2016. We estimated losses of about $11 million US a year from genetic load. We updated those numbers for a paper we just published recently here, and it dropped by almost 3/4. So we're down to about $4 million loss due to genetic load. That's because the allele frequency is so much lower. We have a lot fewer carrier animals, so you're less likely to produce affected calves or embryos. So this is good. This is a success story. We have a lot of genomic information. We're using it effectively. And I have had folks kind of fuss to me, especially when I was at USDA. They would say, well, every time I hear from you guys, it's because you've discovered another genetic defect. And we're sick of hearing about all these new genetic defects. And so, is the number of genetic defects actually increasing in cattle? Like, could there somehow be a relationship between selection and the rate of defects? Well, one of the things we know from some work done in humans is that a lot of individuals carry genetic defects of one form or another. Many of them don't have any effect on the overall health or performance of an individual because biology is messy, right? There's no direction or intention to evolution. So sometimes if you have a gene that's disrupted, there can be another gene that sort of compensates for that. So just because you have a genetic defect doesn't mean you're going to have a problem. And even if you do, the mutations are there even when we can't see them. So what I did is I recently went to the online Mendelian Inheritance in Animals database, and I looked at every published genetic defect in Bos Taurus cattle. And I said, okay, well, when were they discovered? And so that's what I'm showing you on this bar chart here. So the earliest defect reported was first reported in 1893. And then this goes all the way up to like 2023. Because there's a lag between when something's discovered, when it's published, and when it goes in the database. The blue bars are showing, I labeled them phenotypes. This is sort of the classic mode of detection, right? A calf is born, a farmer sees something unexpected, they report it. And then there's sort of this red or maroon is genetic defects that were detected using genomic information. So you can see all the way up till about 2011, we had a pretty constant rate. And I will tell you that over the entire range of these data, it's an average of like 0.96 defects reported per year. Now, in 2011, Paul Van Raden at Agile and our group at USDA proposed a method for detecting genetic defects with genomic information. So that's that first red spike you see. That's because all of a sudden we went back and sifted through all of the genotypes and found things we hadn't noticed before. Basically, all of these are defects that cause embryos to die very early. That's why they hadn't really been detected. It looked on paper like you bred the cow and she didn't conceive. You then see a huge spike in 2013. That's because people all over the world read Paul's paper and then applied the method in their own breed and their own population. And then you can see that things dropped off pretty rapidly after that because we used the new tools available to detect things we hadn't been able to see before, and then everything kind of fell back to baseline because we're not seeing a change in the rate of mutation. Population management is difficult. I won't really go into this. I'm just going to say that on the left is kind of what things look like in the US today. We don't really have a centralized, uniform system for the reporting of genetic defects. There's lots of different points of contact between farms and interested individuals, whether it's through AI companies, veterinary medical diagnostic laboratories, whatever. And then we published a paper recently where we've proposed, and we're working with some industry colleagues, we've proposed a more organized system to mirror what Canada is doing and what some of our colleagues in Europe have done, so that we can better manage genetic defects. So where does this bring us? I'm a geneticist, so I'm going to wrap things up by going back to Jay Lush, right, who was the father of modern animal breeding, an Iowa State professor at Iowa State for a very long time. So he points out in his book Animal Breeding Plans that selection is good for changing genetic merit. It's not really a good tool for managing diversity in the population, and I already said that. And he goes on to further say that if we're successful as geneticists, what we're really trying to do, whether we say so or not, is we're trying to match up the best alleles at each locus in the genome, right? We want to find the best alleles and put them all together in a single animal. And if we're successful at doing that, the resulting animals might be almost completely inbred. So it's not inbreeding in and of itself that's the problem. It's when we do intensive inbreeding without an ability to control where we're doing the inbreeding or the effects of that inbreeding on the population when we build up harmful defects that cause the problems. And we have a few minutes for questions, and I've got some time, so I don't have to jump off here or anything, and I'd be happy to answer questions or explain something that I didn't do a good job of explaining the first time through.
Dr. Cole, thank you very much. A lot of information shared, a lot of great explanations given. We appreciate that. I'm going to turn the first question over to my boss, Dr. Gail Carpenter, who's joined us. What have you got for questions in the queue?
Well, it looks like we have quite a few questions popping up over in the chat, and I encourage anybody that has questions to do so. The first one on there is from Dr. Hansen. It looks like maybe a bit more of a comment, and it includes a lot of math. So I might let Dr. Hansen himself pop up if we need more clarification. But he noted that the estimate of annual increase of average inbreeding of females of plus 0.25% is quite a bit less than what is provided by the CDCB. So I don't know, John, if you had any feedback on that, or Dr. Hansen, if you wanted to clarify or provide more context on that a little bit more. but that was mentioned in the chat there.
Well, I mean, my comment would be that I would trust Dr. Hansen's recall better than mine. Sometimes I get a number lodged in my head, and it's a reminder I should probably double check that to make sure it still reflects the contemporary rate of inbreeding. It will be a little bit different depending on which group of animals you look at, but and I think that actually sort of emphasizes, right, that as inbreeding accelerates, we're going to see an erosion of progress when you compare the increase of inbreeding depression versus the decrease in phenotypic, the decrease in gain from genetic selection.
Dr. Coltis says it seems like with inbreeding, we end up with problems when there is a physiological effect in animals that we don't know about. Are there other traits that we do not measure today that we should measure to be another way to track the true impact of inbreeding that might be faster to obtain and signal problems than, say, survival or fertility traits?
Well, the thing with survival and fertility traits, right, is there's a time lag. So there can be potentially years between the effect of increasing inbreeding depression and what you see. So you could look at calf health traits, for example. I see Colin Lynch is on the call. Colin did a lot of work on calf health traits when he was at University of Guelph, and now he's with Lactanet. Our colleagues at Zoetis have been publishing genomic evaluations for cow health traits in the US for a few years now, and we anticipate at CDCB launching calf health traits for scours and for respiratory disease in December. So probably I would look there first, right? Look something earlier in life so that there's a shorter lag between the expression of the phenotype and so that you can catch things earlier.
Ollie asks, would some limited outbreeding with other breeds of dairy cows be an option for the Holstein Friesian population?
Well, I mean, it's certainly an option, and I think that we saw there's still some very large dairies, right, that are still using a lot of crossbred animals, and that's sort of one way they manage that. But I think a lot of folks, some folks are very comfortable with crossbreeding. Some folks prefer to work with purebred cattle. And so crossbreeding will work, and you can always come back to Holsteins, right? Just because you crossbreed for 1 or 2 generations doesn't mean you have to crossbreed forever. But I don't know. I think we've seen a fairly steady level of crossbreeding in the dairy population these days. So it seems that the folks who are most interested in going that direction have gone that direction, and a lot of other folks haven't taken it up. So yes, it will work. And I know Dr. Hansen and his colleagues and students have done a lot of work showing the benefits of crossbreeding. It's just a different way. But as I commented earlier, if you're going to use crossbreeding, someone still has to maintain the purebred populations. And the folks who maintain those purebred populations are also themselves going to care about inbreeding, or need to care about inbreeding, to maintain those purebred populations, in good health.
Yeah. All right, we have one more question from Ellen. And again, I encourage anybody who's still on to, if you have questions or comments or other feedback, to leave them in the chat. But Ellen is curious about PTAs. So what research are the EFI adjustments to the PTAs based on, and is that updated regularly?
The expected future inbreeding, yes, that is, we just had a base change, right? We do a base change every 5 years, a genetic base change. When we update the genetic base, we also update the group of animals used as the base for expected future inbreeding. And I think that was more noticeable here in 2025 than it has been in the past, right? So we actually had a smaller adjustment for EFI than we have in the past, and that made the base I'm sorry, it was a bigger adjustment, and that made the base adjustment not look quite right. So it is updated. The EFIs are recalculated for every animal 3 times a year. And you asked what research it's based on. Our colleagues at USDA did work on that. I think they published it 2006, 2007. It was some work published in Interbowl Bulletin. And the idea there is that you compare, say, a bull, for example, right? You calculate that bull's relationship to a group of cows selected at random from the population, right? A group of animals in a certain birth year range. And if that bull has a higher than average relationship, he's going to produce more inbreeding than average because of the relationship to the animals in the population. So you would penalize his PTA. If he's less related on average, he would produce less inbreeding in the future if he's made it at random to the population. Now, I mean, to not go too much in the weeds, part of the challenge is that methodology is based on some assumptions about how bulls are used in the population, right? And we really don't see bulls are not randomly mated to, or bulls are not mated uniformly across the population. So it is possible that the assumptions we use when calculating EFI don't exactly match how the bulls are being used today. But there's only, a model. A model is a useful tool, but it's not a perfect tool. So.
Yeah, yeah. And I know that you're coming here as a representative on the Council of Dairy Cattle Breeding, but Kathy does also note that there are defect issues in dairy goat breeds. Do you have any experience with dairy goats, or know any issues?
I'm afraid that I don't. I'm sorry.
Yeah.
I mean, sort of biologically, it's probably they work in broadly speaking similarly to how they work in cattle, and it's the same general consideration, right? If you've got bucks that are very popular, so that they have a lot of offspring in the population, so that their inbreeding is increasing, you kind of have that risk of matching up two undesirable alleles from either side of the pedigree.
Yeah.
But we do not. CDCB does do breeding values for dairy goats as well. We do not adjust the buck PTAs for expected future inbreeding, I don't believe.
Yeah, one more question that we know in the Holstein breed, there are some bulls with a very low coefficient of inbreeding. How good does one of those outcross bulls need to be considered to bring in to your breeding battery?
Well, I think that the key, and tell me if I misunderstand your question, right? The question is maybe really how likely is it that an outcross bull that has a relatively low coefficient of inbreeding because they have a relatively low relationship to the breed, how likely is it that one of those bulls is going to rank high on the list of elite bulls? And it's hard for me to put a probability statement on it. It's going to be hard. And part of that, which I touched on earlier in the talk, is that the statistical methods we use to calculate genetic evaluations really emphasize within family information, and you don't have as much of that for the outcross bulls. And you have a little bit of a chicken and egg situation as well, because when genomic selection was first implemented in the US, several of the genetics companies went out and identified outcross bulls using the genomic information, right? And they offered semen from those bulls for sale, and they sold very little semen from those. And I think most of the genetics companies have really quit looking for outcross bulls, because the fact is they're not competitive against the top bulls in the breed, produced through very intensive family-based selection. So it's one of those things. It's possible, it's just not very likely.
But if you could find one of those that was in the top 5%, you would have a really good slot machine going for you, right?
I would sure think so, you know, especially if you worked with someone who was really interested in selling that bull and really advocating for the merit of using an outcross bull like that. Yes. Yeah. It might have to be top 1% more than top 5%, but that's just kind of the numbers game.
Ellen did have a question. She wanted to clarify, it looks like, her question about EFI. She says she's wondering more about the value of each percentage of EFI. If I remember correctly, each percent point is equivalent to about $25 of net merit. How do you determine how much each point is worth?
Okay. I think, without breaking out of my presentation and going digging through stuff, I think it's closer to $40 now per percentage point. So the way we get that number is, I've calculated this before. I look at the inbreeding depression, right? So the change in performance for a 1% increase in inbreeding, and then I multiply that. So let's say it's 140 pounds or whatever of milk. I multiply that by the value of milk that we use to calculate the net merit selection index, and then I add that up across all of the traits. So basically, it's an average that reflects the value of the production gained or lost from the change in inbreeding. So I hope that answered her question better than I did the first time.
Les would like to know, isn't an additional concern that as genetic diversity within a breed decreases, that it becomes more challenging to make genetic improvement?
Yeah, and Les, if I misunderstand your question, please let me know, right? Because I feel like part of this is the question is, at least in theory, as inbreeding increases, right, you see a decrease in genetic variance in the population. So long term, yes, it gets harder to maintain, to make as much genetic progress. What I can say is that when we've looked at that with the data we have available to us, we haven't really seen much change in the rates of genetic gain or in the estimates of genetic variance. So today, that's not happening. In the long term, it's quite likely that yes, we would, not only would we see more loss due to inbreeding depression, we would also not be improving as quickly. And so you sort of get a double, you get hit from both directions. So I think, Les, I'm agreeing with you, and I are in agreement, I believe, on this. Now, the question is really, it's a little bit like the more general question about inbreeding. When will we get to the point where we see a big change in the realized genetic variation in the population? We're not there yet. Thankfully, we don't really know. Now, maybe I'll say, though, that I'm not trying to give the impression that I think everything is fine and no one needs to change what they're doing. Inbreeding is kind of a classical collective action problem, because a lot of people all have to work together towards a common objective to change the situation, but each person has kind of an individual incentive not to do that. And I don't really know how we effectively change that absent some more central authority, whether it's a breed association saying that we won't register animals that exceed a certain coefficient in breeding, or if the customers of genetics speak with their dollar and say, you know what, I'm very concerned about inbreeding, and I'm simply not going to purchase semen from bulls or purchase embryos that continue this rate of inbreeding. I don't think we're very likely to see any sort of governmental intervention for a number of reasons. But I'll be honest, I know some folks who are crossbreeding feel like they don't have to worry about this at all because I just crossbreed. So what do I care about inbreeding? Well, you'll care about inbreeding when you're not able to make the progress you want to make because the purebreds aren't improving at the rate that you would like. And again, I don't really see gene editing in some of these tools as any kind of magic fix to that.
We have a comment from Dan Bolin that it's line breeding when it works and it's negative. When it's negative, it's inbreeding. And I'm sure we've all heard that before. Yep.
That is as accurate a thing as I've ever heard. Yes.
With that, I believe we will thank Dr. Cole again. We appreciate the energy and the information you brought to this program. And thank you. We will go ahead and terminate now and invite everybody to come to the program in June, where we will be talking with the folks, the herd that wins our 7-pound contest up at the 4th State Dairy Nutrition Conference. So thank you again, and we will see you.
Yeah, I just like to say I appreciate everyone taking the time to listen today and to ask questions, and hope everyone has a good day.
Thank you now.
Yeah, bye-bye.